Hi, everyone. This is Professor Kennedy Edem Kupia. Once again, I bring you another lecture in pharmacology. And today we want to look at tetracyclines. Now, what are tetracyclines? From the name, tetra means four. And so these are a group of broad spectrum antibiotics that have four fused cyclic rings. So when you look on the board, you'd realize that the structure I have in the corner, there is one cyclic ring here, there is a second one here, there is a third one, and then there is a fourth one. So we have four cyclic rings that are fused. So it is from this four cyclic rings that are fused that we get the name tetracyclines. Originally, these drugs were isolated from other microbes. In fact, they were isolated from soil actinomycetes, but others were produced by modifying the structure of the natural ones. Now, because we describe these drugs as broad spectrum antibiotics, this comes with some implications. The fact that they are broad spectrum antibiotics would mean that these drugs have a wide range of microorganisms that they attack. So by attacking some of these wide range of organisms, they are able to clear what we call the normal flora, that is the normal organisms that are resident in your body. Some of these, their function is to protect you against the growth of other pathogenic organisms. So when a drug is a broad spectrum antibiotic, it means that it is likely to clear the beneficial microorganisms that we need. And so one of the challenges that could occur as a result of that is that we may get what we call pseudomembranous colitis. That is one of the challenges that is associated with using broad spectrum antibiotics. Now let's come to some of the examples of these tetracyclines. So the natural ones that we isolated from the microbe, we have chlortetracycline, which was the first one, oxytetracycline, and then tetracycline itself. Then those that came by modifying the natural ones, we have demiclocycline, metacycline, minocycline, doxycycline, rolitetracycline, limecycline, tigecycline, which belongs to a subgroup we call the glycyl cyclines. Then omadacycline, which was discovered by a Ghanaian professor. And we have saricycline. Saricycline is produced mainly for the purpose of dealing with acne, right? Then we have a synthetic one called eravacycline. Now, if we want to understand the pharmacology of tetracyclines, we would want to look at how they do their work exactly. Okay, how do they do their work? Usually we describe tetracyclines as a group of drugs that work by inhibiting bacterial protein synthesis. So how do they inhibit the protein synthesis? Now imagine a tetracycline that has been given to a patient. The patient has swallowed the drug has been absorbed, it has been distributed, and it has gotten to the sites that it needs to work. So this is our tetracycline right here, okay? So let's say that is the site it needs to work. Now let's pick our microorganism. So let's say this is the structure of our bacterium, right? Now, if we pick this bacterium, our interest is on the fact that the drug is going to inhibit protein synthesis. So we are going to focus on what we call the ribosomal subunit and then the mRNA, right? So let's pick this is the structure of our mRNA, has the codons that determine the kind of amino acids that we'll be using to form the peptides and then the proteins. Now, let's assume that this is a part of our ribosomal subunit. We will call this the 50S ribosomal subunit. And then let's give this one 30S ribosomal subunit. Now, these ribosomal subunits more or less come together as a complex. I have separated them so that I could use that to illustrate my point. But they come together as a complex to serve as a factory for the synthesis of the protein, all right? Now, there are three different sites that we would have to look at. We have what we call the A site, we have the P site, and then the E site. The A site is where the acceptor site. So that is where we bring the amino acids when we want to form the proteins. And then the P site is the peptide site. Now, for you to produce proteins, for the microorganisms to produce proteins, they need first amino acids coming together. These amino acids need to come together to form peptides. So by stringing a number of the amino acids together, we form peptides. And by folding the peptides later, we have the proteins. So these microorganisms have to go through this process to form their proteins. So usually we would expect that transfer RNA would bring in the amino acid, and then it brings it to the A site. And then this, as it brings more, we would be forming a group of peptides at the P site where you would have various amino acids. So let's say amino acid, another amino acid, another amino acid. So we will be joining those together to form our peptide. Then as this is happening, once the mRNA shifts, we'll have another codon that will allow us to be able to determine what kind of amino acids to be bringing in. Then the tRNA would leave through the E site to go and bring in another amino acid. So this is how the process continues until we produce the peptides needed for the protein. Now, what the tetracycline does is that when you administer it and it enters the microorganism, it binds to the 30S ribosomal subunit at the A site, as you can see from the animation. So by binding to the A site, it will now prevent the tRNA, that is the transfer RNA, from bringing in any more amino acids. So imagine the bacterium wants to produce a protein that has 200 amino acids, but then it has only produced a peptide with 50 amino acids, and it needs more amino acids to be added, and then tetracycline is given. Once tetracycline is given, it means that we cannot bring in any more amino acids. So the peptide chain cannot elongate, and we will not have the protein we need to have. Now, because protein helps with growth and division, it means that the microorganisms may not be able to grow. This is why we normally say that tetracyclines are bacteriostatic, but at high concentrations, they can be bactericidal, okay? But usually they are bacteriostatic against most of the organisms that they affect. I hope this is simple enough for us. Now, what is their spectrum of activity? We have already said that tetracyclines are broad spectrum antibiotics. So usually they target gram-positive bacteria. This is the activity is normally skewed towards gram-positive bacteria, but they also affect gram-negative bacteria, in fact, including aerobic organisms, anaerobic organisms, some spirochetes, and then rickettsia. They affect all of these organisms. Then they are also able to attack organisms that are not bacteria. So they attack protozoa. So specifically, we have entamoeba, histolytica, which causes amoebasis. Then we also have Plasmodium falciparum, which causes malaria. So the protozoa they attack, these are the two that they normally would attack. Now, based on the organisms that they would normally attack, what do we use the tetracyclines for? One of the things you want to keep in mind is that The tetracyclines used to be very popular years ago, but now their popularity has declined due to what we call resistance, okay? But they remain a drug of choice for various conditions like pneumonia caused by mycoplasma pneumoniae. Then they could also be used in chlamydia infection. They could be used for typhus. Remember, this typhus is not typhoid fever. Typhus is caused by a rickettsia organism, right? So this is typhus, and then they could also be used as drugs of choice in cholera. Then aside this, we could use them together with other medications in what we call triple therapy in dealing with duodenal ulcer that is being caused by Helicobacter pylori. We could also use them in combination with aminoglycosides to deal with plague, tularemia, brucellosis. Then they could also serve as alternative medications in managing amoebasis and in malaria prophylaxis. So assuming someone who is living in a non-malaria endemic region is coming to a place where there is malaria, usually that person could be given tetracyclines to reduce the possibility of being susceptible to malaria. Then we would look at the other situations where we would use them. We could talk about acne. We could talk about, so doxycycline, saricycline, as I mentioned. We could use them in exacerbation of bronchitis, community-acquired pneumonia, and then a syndrome of inappropriate secretion of ADH. Here, demiclocycline is one of the drugs we tend to use in that condition. Then we could use it in Lyme disease and then relapsing fever. In fact, there are others, but I think this captures majority of the things we could use tetracyclines in.